Last week we ran a DNA gel on Site 7 and found a restriction pattern nobody could match. This week we run an SDS-PAGE on the protein fraction from the same samples, the one we extracted in week 2 and stored at -80. DNA tells you what the genome looks like. Protein tells you what it's actually doing. This is SDS-PAGE, and I need to know what's expressing in that drainage ditch. Quick version if you're new. Method: Monday, one technique per week. They build on each other. DNA extraction was week 2. Gel electrophoresis was last week. This week we go from nucleic acids to proteins. Same principle, different chemistry, completely different resolution requirements.
Sodium dodecyl sulfate is an anionic detergent. It binds to the hydrophobic regions of a protein at approximately 1.4 grams of SDS per gram of protein. That ratio is essentially constant across all proteins. The result, every protein, regardless of its native shape, charge, or function, is converted into a linear rod with a uniform negative charge to mass ratio. Native charge is irrelevant. Native structure is irrelevant. Migration through the gel now depends only on molecular size.
SDS costs about $20 for 500 grams. It does more work per gram than almost anything else in a biochemistry lab.
Why polyacrylamide and not agarose? Agarose pore sizes range from about 50 to several hundred nanometers, adequate for large DNA fragments, but too coarse to resolve proteins in the 10 to 200 kilodalton range. Polyacrylamide pore size is controlled by 2 parameters: total acrylamide percentage and the ratio of bisacrylamide crosslinker. You can tune the mesh to match your molecular weight window with precision you cannot achieve in agarose.
It is mildly satisfying that the same electric field principle that moves DNA moves proteins, but everything else about the system is different.
Subscribe. Every Monday, one method that actually tells you something. 2 weeks ago, it was extraction. Last week, it was gel reading. This week, we find out what protein is in a drainage ditch in the eastern sector and why I'm losing sleep over it. Equipment. Gel first. You can hand cast or use precast. Precast gels are consistent and save 45 minutes. If you're running a standard experiment, use them. If you need a specific percentage or a gradient gel, you're casting by hand. I cast by hand because I don't have a reliable precast supplier that ships to a residential address without questions.
I'm using 15%.
For what protein sizes?
The Site 7 extract, something might be at 200 kilodaltons.
A 200 kilodalton protein does not enter a 15% polyacrylamide gel. It stalls at the stacking resolving interface. You will see a smear at the top and clean lanes below 60 kilodaltons. Get an 8% gel.
Polyacrylamide gels follow the same size exclusion logic as agarose, but in a tighter range. At 15%, the effective separation window is roughly 10 to 60 kilodaltons. A 200 kilodalton protein cannot reptate through that matrix. Unlike in agarose, where you can sometimes recover from a concentration error by running longer, in polyacrylamide the protein genuinely cannot enter the gel. You recast.
It is mildly consistent that every gel technique has the same failure mode when the wrong percentage is used.
Precast gels come in standard percentages. Hand casting lets you tune. If you're paying for precast gels and picking the wrong percentage, you are paying for the convenience of wasting the experiment faster.
Running buffer. Trisglycine SDS, the Lamley system. Make 10x stock, dilute to 1x before use. The SDS in the running buffer maintains the denatured state during migration. If you run without SDS in the buffer, proteins may partially refold during the run, and your bands become meaningless. I've done this. The gel looked like a mistake. It was. Loading buffer. 6 times Lemley, SDS, TT or beta-mercaptoethanol, glycerol, bromophenol blue, tris pH 6.8. The reducing agent is not optional. It breaks disulfide bonds. Without it, proteins that are linked by disulfide bridges migrate as intact complexes at the wrong molecular weight.
I made loading buffer this morning. I didn't have beta-mercaptoethanol, so I left it out.
Do you have DTT?
No.
Then every protein in your sample that has a disulfide bond is going to run at the wrong molecular weight. If it's a disulfide bonded multimer, it won't separate into subunits. You'll see one large band where you expect several smaller ones and nothing where those subunits should appear.
Disulfide bonds between cysteine residues hold protein quaternary structure together in oxidizing conditions. Beta-mercaptoethanol and DTT both act as reducing agents. They break those bonds, freeing each polypeptide chain to migrate independently. Without reduction, a disulfide bonded dimer migrates at twice its monomer mass. The apparent molecular weight is wrong, and the error is not obvious from the band pattern alone unless you run a parallel non-reducing lane for comparison.
It is mildly concerning that the component most frequently left out of loading buffer is the one that determines whether the molecular weight data is correct.
Protein ladder. Pre-stained ladders are visible during the run and let you track migration in real time. Choose one that covers your expected range. If you're looking for something at 200 kilodaltons, use a ladder that goes up to 250. The same rule as DNA. Extrapolating off the ladder is not science, it's guessing. Step 1. Sample preparation. Mix sample with loading buffer. Standard is 1:1 with 2x Lemley, or 1:5 with 6x Lemley, to keep volume low. Boil at 95 degrees Celsius for 5 minutes. Boiling is required. SDS alone at room temperature doesn't fully break secondary structure. The heat completes the denaturation. After boiling, spin briefly, 1 minute at 10,000 gauges, to pellet any debris. Load the supernatant.
I skipped the boiling. I thought the SDS would be enough.
It isn't. At room temperature, SDS disrupts hydrophobic interactions but leaves some secondary and tertiary structure intact. Your proteins will run at partially native molecular weights. Bands will appear at positions that don't correspond to actual molecular mass. The gel will look like it worked. It will be wrong.
Complete protein denaturation requires heat because the unfolding of alpha helices and beta sheets is an endothermic process. The SDS protein interaction at room temperature is insufficient to drive full chain extension. At 95 degrees, the combination of thermal energy and SDS ensures complete linearization. 5 minutes is empirically validated for most proteins. Glycoproteins and some membrane proteins may require longer.
Step 2. Load the gel. 10 to 30 micrograms of total protein per lane is the range for Kumasi staining. Less and you won't see faint bands. More and your lanes smear. Pipette slowly into the wells. The stacking gel wells are small and fragile. Load your ladder in the first or last lane, or both.
The protein concentration range matters because Kumasi staining has a limited dynamic range. Below roughly 50 nanograms per band, intensity is below detection threshold. Above a few micrograms per band, the stain saturates and bands lose their sharp margins. The local SDS concentration is too high, electromigration becomes non-ideal, and adjacent bands compress together. Bradford or BCA assay your samples before loading to know what you're putting in.
Bradford reagent costs about $3 per sample. Running an SDS-PAGE gel on an unquantified sample costs 30 minutes of runtime plus staining time plus the possibility of having to do it again. Quantify.
Step 3. Run the gel. Start at 80 volts while samples are in the stacking gel. The stacking gel concentrates all proteins into a tight band before they hit the resolving gel. You need lower voltage here to let the Kohlrausch discontinuity do its job. Once the dye front enters the resolving gel, increase to 120 to 150 volts. Run until the bromophenol blue front reaches the bottom of the resolving gel. Total runtime is usually 60 to 90 minutes.
The stacking gel operates by a discontinuous buffer system. The stacking gel is pH 6.8. The resolving gel is pH 8.8. At pH 6.8, glycine in the running buffer carries minimal charge. It migrates slowly. Chloride ions from the tris-HCl buffer are fast. Proteins become sandwiched between the slow glycine and the fast chloride, compressing into a razor-thin band at the stacking resolving interface. When that band enters pH 8.8, glycine becomes fully ionized, overtakes the proteins, and the size-based separation begins. This is the Kohlrausch discontinuity. It is why SDS-PAGE gives sharp bands rather than diffuse smears.
Wait, if every protein starts in a single compressed band at the top of the resolving gel, how do they end up separated? Don't they just migrate as one compressed block?
Once they enter the resolving gel, each protein's migration rate is determined by its size and the polyacrylamide pore size. There's no longer a driving force keeping them together. The stacking is a starting condition, not a permanent one. Separation begins immediately as the band crosses into the resolving gel.
Step 4. Stain. Kumasi R250 and methanol and acetic acid is the standard. Fix the gel first. 30 minutes in methanol acetic acid solution. Then stain for 1 hour. Then destain. And this is where most people ruin an otherwise clean gel. They don't destain long enough. The background won't be clear at 1 hour. It won't be clear at 2 hours. Leave it overnight. In the morning your bands are sharp and your background is clean. Rushing destaining gives you a blue rectangle with darker blue rectangles in it. Technically a gel, not useful.
Kumasi interacts nonspecifically with the polyacrylamide matrix as well as with proteins. The destaining step removes the unbound dye by diffusion into methanol acetic acid solution. Diffusion is slow. The time constant is hours, not minutes. The protein-bound dye is retained because the protein-dye interaction is stronger. You cannot accelerate this without heating, and heating distorts the gel. Patience is the protocol here.
Good gel. Discrete sharp bands at expected molecular weight positions. Your ladder bands should be tight and evenly spaced when plotted on a log scale. Your sample lanes should show bands that correspond to the known proteins in your preparation, with relative intensities that make sense for relative abundance. If you're running a crude lysate, you'll see many bands. If you're running a purified protein, you should see one or two if it has subunits.
A log linear plot of ladder molecular weight against migration distance gives you a standard curve. Read your unknown bands off that curve to estimate molecular weight. The relationship is linear in the middle of the resolving gel, but breaks down at the extremes. Very large proteins near the top and very small proteins near the bottom migrate non-linearly. Your sizing estimates are most reliable for bands in the middle 60% of the gel. Two distinct failure modes produce similar-looking gels. Overloading, more than about 30 to 40 micrograms total protein per lane for Kumasi, causes bands to compress laterally. The SDS protein, micelles, become too dense for independent electromigration and bands bleed into adjacent lanes. Underdistaining produces a high background that obscures faint bands. In both cases, the gel looks messy. The distinction overloading smears the bands themselves. Underdistaining leaves a uniform blue background with sharp bands partially visible beneath it.
It is mildly clarifying that a bad gel tells you whether you added too much or waited too little.
Closing tip on reading the gel. You are not estimating molecular weight by eye. You are building a standard curve, log of each ladder, bands molecular weight on one axis, migration distance in millimeters on the other. Draw the line. Read your unknown bands off that line. If you're doing this in a published experiment, do it quantitatively. Approximately 50 kilodaltons is a statement you make after measuring, not instead of it. Write the Site 7 protein fractions. I ran 3 collection dates from Site 7 alongside 4 control sites. Used ripolysis buffer, which we covered in week 2, because I needed denaturing conditions for direct SDS-PAGE compatibility. The control sites gave expected profiles. Housekeeping proteins, a clean ladder, nothing surprising. Site 7 has a band at approximately 47 kilodaltons that doesn't appear in any control site. It's not a housekeeping protein. It's present in all 3 collection dates. It's bright enough on Kumasi to suggest it's one of the most abundant proteins in that sample. That's not a background artifact. Something is expressing a 47 kilodalton protein in high concentration in that drainage ditch, and it's been doing it consistently for 8 months.
I ran silver stain on the remaining gel. The 47 kilodalton band is still the brightest thing in the lane. The control sites don't have it.
It is mildly significant that the most abundant protein in the sample is the one nobody can identify.
A highly abundant unknown protein at a consistent industrial site. I'm not saying bioremediation. I am saying that whoever finds out what this is first is having an interesting conversation.
Closing tip. Don't run at constant current. Run at constant voltage. At constant current, as the gel heats up, the resistance drops, the voltage rises, your bands blow apart, and your separation deteriorates in the last 20 minutes of the run. Exactly when you need the most resolution. Constant voltage, 120 to 150 volts. The run takes a few minutes longer. The bands are worth it. That's SDS-PAGE. Denature your proteins. Stack them into a sharp starting band. Separate by size through polyacrylamide stain and read the gel. The Site 7 47 kilodalton band is consistent, abundant, and unidentified. Next Monday, Western blot. We're going to transfer that gel to a membrane and probe it with an antibody. If I can match the protein, we'll know what organism is expressing it and why it's concentrated in an industrial drainage corridor. Method Monday. Every Monday.